There we go.
Okay.
Sorry about that, everybody. Let me just back up here. So here's our agenda for today, just so that we have it recorded for posterity's sake. So let's talk about who Advaita is. So we're a team of 8. We've got a couple of additional people that are joining us. We have these grants, and you can see a list of some of the customers that we have currently. So the iPathwayGuide application is really designed for looking at protein and gene expression data and give you that tertiary analysis, and I'll explain what that means in just a minute. But basically, we have an analysis of what are your DE genes. We predict the active microRNAs. We do a gene ontology analysis, as well as a pathway analysis with the ability to model drugs, microRNAs, and SNPs. We also have the ability to identify enriched diseases, and we also have a meta-analysis feature. And I'll go into each one of these in the live demonstration. So some of the key features and benefits of iPathwayGuide are the method, and I'll explain in detail what that method is, that really helps drive significant results or meaningful results that you can take for your next experiment. We also have a very flexible pricing and licensing model, which helps to reduce your costs. And we have a very easy to use interface, which helps to shorten that learning curve. So you don't have to be a bioinformatician in order to use iPathwayGuide. There are a few other features and benefits that you can read there, but those are, I think, some of the key things that we like to highlight for iPathwayGuide. Here's a quote that we recently received. A highly renowned collaborator proclaimed I had given him the bioinformatics analysis he had been hoping to get for 20 years or so. My aim is to provide a set of testable hypotheses to clients, not simply a list of genes. Your software fits that bill extremely well, in my opinion. And we've heard this more than once from a number of bioinformatics cores, and I think this is a testament to the type of analysis that you can get with iPathwayGuide. So let's talk about the pipeline that most expression analyses go through. So you start off on the left here with your instrumentation, and you get some, let's say, RNA-seq or protein expression. You do some alignment and annotation, and then you have your 2 phenotypes that you want to compare. So you have your control versus your comparison group, and you have your differential expression. Then you have your tertiary analysis, which is your pathway analysis, and that's really where iPathwayGuide fits in. We do a little bit of differential expression analysis on microarray files, but predominantly we take the differential expression data as their input, and we provide you that pathway analysis. We also have a quaternary type of analysis where you can compare multiple pathway analyses against one another and elucidate what are the key drivers between those different experiments. So let's talk about some of the classical approaches to pathway analysis. The most traditional method, and it's used by the other commercial packages that are out there, is overrepresentation or an enrichment type of analysis. And it's a really simple type of analysis because you don't need a lot of data in order to run it. The detriment, though, is that this type of analysis assumes that the genes are independent, and we know that the genes are not independent. In fact, they're part of systems. That's what these pathways are there to describe, is those relationships between those genes. So an improvement on that overrepresentation analysis was this functional class scoring, which does a little bit better job, but it still assumes that the genes are independent. So then a method was proposed for taking the topology of a pathway into account, looking at the interactions between the genes and consider the role and position of each gene, and that seemed to be kind of that next generation. Well, what we've done is we've combined both the overrepresentation analysis as well as the pathway topology analysis. These are orthogonal measures, so we can compare these together and basically look at 2 forms of evidence in order to assess the relevant impact of each pathway. So the method was published in Genome Research in 2007. You can see the citation here at the bottom, but there's real science here behind it. Basically what we do is we look at all of the upstream genes and all of the downstream genes, and we assess the relationships that exist between them, whether it's 1 to many or many to 1, and we are able to look at all of the downstream perturbations in that particular pathway. And let me give you a real world example here in just a second. But when we plot the scores, we use the overrepresentation analysis or enrichment on the horizontal axis, and then we plot the perturbation on the vertical axis. And these two forms of evidence, if they're both significant, you'll end up with pathways in this top right corner, and those are the ones that are going to give you the most significant results. So here's that real world example that I was talking about. This is the insulin signaling pathway, and if we just assume for a second that we had just one DE gene, the insulin signaling receptor in this case, and we wanted to assess the relative enrichment, we would see that this is just one of maybe 50 genes on this pathway, and that's all we can really assess is how likely would we see one gene out of this many genes by random chance alone. Whereas with our perturbation analysis, we're going to look at the position of this particular receptor, and we're going to see that this is the main entry point on this pathway, and that it has several downstream components. And so if this gene is perturbed, we should see a downstream effect of that gene throughout this entire pathway. And so we would be able to assess that this pathway is quite impacted. When we look at the adherence junction pathway, we have that same insulin signaling receptor here, and this is one of several entry points on this particular pathway. Again, from an enrichment standpoint, this is just one of many genes on this particular pathway, and that's all we can assess from that. But using our impact analysis method, we would see that this particular entry point is not that connected on this particular pathway, and therefore we would score this particular pathway lower than the insulin signaling receptor pathway. So let's talk about what is required for input and what you get for output with iPathwayGuide. So the input is the entire list of genes, and I want to stress that other packages will allow you to just submit just DE genes, but because we want to look at what the relative background is, we would like to have the entire list of genes submitted along with their associated fold changes, and then p-values. We recommend that you use corrected p-values, or sometimes referred to as q-values in certain workflows, and it just generally tends to give you better results. The output will be a list of DE genes, Go terms, predicted microRNAs, diseases, and impacted pathways, as I had previously mentioned. The organisms that we currently support are human, mouse, and rat. And the file formats you can see here that we support, basically, if you have differential expression, we can analyze it. So I've talked a little bit about the features. We're going to go into a live demonstration here in just a second. Just in case we do run out of time or run short on time, I did want to point out a couple premium features that we offer, one of them being the Metaanalysis. This is where you can build and compare up to 5 separate data sets, and you can even compare multi-omic data sets. So if you have a proteomics data set and a transcriptomics data set, you can compare those against each other. The other premium feature, and I think this is one of the things that is a huge win for larger labs as well as core facilities, is this printable report. This is an automatic generated report, and I'll show you an actual one here in just a minute, but it's about an 18 to 20 page paper that's automatically generated based on your data, and can be used as supplementary material for either publications or grant applications and the like. So with that, I'm going to dive right into the application demo. And let's see here, it's a simple login. And I noticed that most of the people had already had accounts established with us, so I'm not going to go into the registration process. But if you do need help, feel free to contact us, and we'd be happy to work with you on getting registered. So I'm going to do a very simple upload of some new data. We're going to have a separate webinar on the different types of data and how to upload them and get them in, but I just wanted to go through the very basics. So I clicked on Create New Report, and we choose our organism. I'm going to use Homo sapiens here. You have a variety of file formats that you can choose. I'm going to use CuffDiff, and then we just choose the file that we're going to upload, and In this case, I have a cuff diff file, so this is part of the top hat and cufflinks workflow that a lot of core facilities use. And this is also a workflow that's available in base-based. And I suppose I should mention, I'm going to be demonstrating the native version of the application and not the base-based version. We'll be doing a base-based centric webinar in the future. So in this case, our cuff diff file had 3 comparison groups. So one is selected by default, but I could choose which ones I wanted to analyze. You can choose up to 5. I just want to give my report a quick title, whoops, and a description. And then if I wanted to, I could change my thresholds here, but you can see that each one of these data sets has roughly 11,000 genes that were measured. We have a fold change cutoff of 0.6 and a p-value of 0.05. These are the standard thresholds. And you can see that I have, based on those thresholds, I have 214 genes that were selected as DE genes. If I wanted to change this maybe to 0.3 for my fold change, I would simply enter that and hit view and apply. Excuse me. And you can see that the number of DE genes has updated to now 235. And we also have a preview of our volcano plot down here. If we're satisfied with this, then we would just simply hit Analyze Data, and this will zoom off into the cloud, and we'll see that this is pending, and then we should get a result in about a half an hour or so. Generally, that takes about 15 to 30 minutes for an analysis to complete. You'll get an automated email once it is completed, so you don't have to sit here and baby it. We'll send you that email. So on the dashboard, you have 4 different categories. We have purchased, we have expired, we have trial, and we have pending. So the way that iPathwayGuide works is that for each data set that you upload to us, if you don't have a subscription, each of those data sets is available for 72 hours for viewing, and you can look around and click on all aspects of the result. Obviously, downloading is not enabled because otherwise we wouldn't make any money. And then after 72 hours, it goes into the expired category. It's still there, and you can still purchase it after the fact if you so desire. Or you could just move it into your trash bin, and then you don't have to look at it any further. So while that's processing, I'm going to pull up a public data set that does a really nice job of highlighting the power of iPathwayGuide. So this is comes from a public data set. So this is GSE 47363, and you can look this up in NCBI Gene Expression Omnibus. And here they have a classic treated versus non-treated type of phenotype comparison where they have a cell line, and they treated that cell line with a microRNA mimic, and they wanted to see what would be the effect on gene expression. Before I go into the actual results, I just wanted to let you know that when you have an analysis and it's completed, you can click on the title and it'll open up information here, so you can edit your report title or your description. You can even change the contrast labels, but you also have information about the pathways and the other databases that are used in the analysis, when they were updated, and when we provide an update to the database, a button will pop up here that will allow you to rerun your data on the latest data. We will preserve your existing results, so those will always be there based on the data set, the original databases that it was run against, but you can also update it to the latest biology. So we'll just close that. So this is the summary page, and the way I like to look at this is our input over here on the left, and on the right 2/3 is the output. So here we have about 20,000 or almost 21,000 genes that were measured. We have about 1,200 that were significantly differentially expressed using our default thresholds of 0.6 for the fold change and a p-value of 0.05. On the output side, we have the top biological pathways, predicted microRNAs, top gene ontology terms, and top diseases. Now you can certainly navigate to each one of those things directly from here. You can see that it changes to a link, but I'll go through each of the tabs individually just to take you in more detail. Here's that printable report that I had mentioned before. Clicking on that on a purchased report will automatically generate this paper for you. And here you have what is the input data, what were the thresholds that were used. You have the detailed method section here, including the formulas that were used. And then we get into the actual results, uncorrected and corrected, with figures and captions and all kinds of stuff that most people would find very useful in this type of a paper. We even go so far, if I zoom down to the bottom here, we even provide a references section for the references that went into the creation of this analysis for you. So a lot of the legwork is already done for you in that case. All right, so from the summary page, what we're going to do is march across these tabs at the top. And I just realized I should probably be in presentation mode. So we'll go into the genes section here. So here we have a, I would call it a gene browser based on your data. And the way that this works is the volcano plot here is clickable. You can click on any one of the genes and get updates on that particular gene that you might be interested in. Similarly, if you want to search for your gene of interest, you could type it down here and say you're interested in MAPK. You would click on that. It pulls up relevant information about that gene, but then you can also get into what are the biological processes with this gene annotated to it, or molecular functions, or what pathways contain MAPK, as well as any drugs that are known to target that, and microRNAs and so on and so forth. So you get an idea, and this will become more relevant as we go through the demonstration here of how you can really leverage this section of the analysis. One thing before I go too much further, you'll see this little download arrow, and when you click on that, you can save the plot either as a PNG or an SVG file, but the table below it, you can save that as a CSV. And so that can be very useful if you wanted to get a custom view of or a custom type of graph for your reporting needs. We also have these little camera icons, and you'll see these sprinkled throughout the application. When you click on that, a little video pops up. I won't play it here for you, but basically it's a 1 to 2 minute video that provides you with just-in-time education for that part of the analysis. So you don't have to sit through this webinar once again to distill those elements. So it's a nice little thing that just keeps you zeroed in on that part of the analysis. So let's go on to the microRNA analysis, and this is the first real analysis in the application. So we provide a prediction of what microRNAs may be active based on the gene expression signature in your data that you've uploaded. So by default, we do not provide a correction factor, but we can apply, say, Bonferroni, which is the most stringent that we offer. And you can see that the table is updated, and currently we have 4 candidates that may be significant or may be active. And this top one, this MIR542-3P, we can see is 10 to the -4. So this is a highly significant result that we were able to get. And the way that we read this is that of the number of DE genes that are targeted by this particular microRNA, we have 45 are downwardly expressed out of 47 that were DE. And we have that modeled over here on the right-hand side, where you can see each of the genes expressed in a bar plot, and here's our 2 that were not downwardly expressed. Here's the cool thing about our application, or one of the cool things, is that this is all interactive. So if I wanted to learn a little bit more about this ILK, this integrin-linked kinase gene, I can click on that and find out more information about that. So it's meant to be very interactive, and even during the trial period, interactivity is available to you. So the really cool thing, as I mentioned here, we predicted here that this miR542-3P is the most likely candidate for a microRNA. It happens to be that was the microRNA that they used to stimulate this cell line. So very quickly we've identified a likely candidate for that microRNA, and it would be a great hypothesis generator so that we could go and do, say, a microRNA expression experiment and identify is indeed that microRNA active in this particular sample. Okay, so let's move down to the gene ontology analysis. And gene ontology is a structured vocabulary for describing one of three domains. We have biological processes, cellular components, and molecular functions. I've chosen biological processes in this case, and the analysis is the same for all three domains. The way that we read this graph is we start at the 12 o'clock position, work our way counterclockwise to the most significant. So cyan, or this light blue, is the least significant, and the magenta is the most significant. Again, by default, we don't apply a correction factor. For ontologies, because it is a hierarchical relationship, we provide these advanced pruning algorithms. So we have weight and elim pruning, elim being the more stringent. And there's a paper, if you look on our FAQ that, or in our user guide, that links to the paper that describes these. And then we can also put a threshold here of how many DE genes do we want to have annotated before we would consider that term. So here I've just randomly selected 22 DE genes, and you can see that our graph has quickly narrowed down to a list of likely candidates. So I've chosen the most significant one here, which happens to be the negative regulation of cell proliferation. So this is 10 to the minus 8 significance, and you have a little bit more information about this particular Go term. And you have a link out to the ontology term itself, but then we have the bar graph that of the annotated genes to this particular Go process right at our fingertips. Below that, we have the ancestor chart. And the way that these ancestor charts are read is we have the most specific term. So this is our negative regulation of cell proliferation term. And then it gets more general the further you go up. And in this case, in the biological processes, they will all terminate with biological process as the root gene ontology term. And of course, on the right here, we have a legend so that as you highlight over each of these Go terms, you can see what are the relationships between these various gene ontology terms, and you can read that accordingly. If you wanted to collapse the tree, say, down to a particular term, you can click on this little gene ontology number in the header, and then it'll collapse the tree down to that specific term with the DE genes that are annotated to that one. So, and again, this is all downloadable. If you wanted to, you could download this as a PNG. Same with the bar graph. You can download this either as a PNG or as a CSV table and include that in any type of paper that you may be working on. So that's the gene ontology analysis. Let's move on over to the pathways analysis section. So if you remember, we use two forms of evidence, and this is the key to our impact analysis, where we look at the p-value from the overrepresentation analysis or enrichment on the horizontal axis, and then on the vertical axis, we look at the p-value from the accumulated perturbation or the topology-based analysis. And in this case, we have the pathogenic E. coli infection pathway as our most significant pathway. Again, we don't have a correction factor applied by default, but we can apply an FDR and narrow our list of results. In this case, we have the E. coli pathway as being 10 to the minus 6 for significance. On the right-hand side, we have the pathway diagram, and then below it we have the DE genes that are annotated to that particular pathway. So by default, we are showing the log fold change of the p-values that came from our expression experiment, whether that's a gene expression or protein expression. And some of these on these pathways are complexes. So here we have a tubulin complex, and you can see specifically which genes in that complex were measured, and you get a sense of what their relative fold change was based on the scale up at the top. So as we mentioned, we look at how the genes are connected in this particular system. So here we have the fold change, but we know that the pathway describes that there is a relationship, a downstream relationship. So when we assess the topology, the downstream component of that, by looking at the total perturbation accumulation, that's the downstream accumulation of this fold change, we can see that we have a number of genes that are predicted as having some type of change in expression. So when we look at the total system perturbation, we look at what was measured plus the downstream component of that. And I know that's a lot to take in this short webinar, but if you have any questions, feel free to just click on the video camera in the top right corner and you'll get another explanation of it, and you can pause it and kind of digest it as needed. Another nice element that we have is this ability to show coherence in the pathway, or coherent cascades. And what this is when the measurements in the pathway, as well of the genes in the pathway, are in accordance with the relationships as described by the pathway, then we highlight those, and these can serve as putative mechanisms in the pathway. So you would want to focus in on these areas for hypothesis and for maybe going on to your next experiment. So we take a lot of the distillation of these data out of just make it easier for you, and just so all you have to do is click a button, you can look at what's really important to you. And then on the right here, we can look at the pathway details. So I'm going to expand this panel here, and we have a number of additional visualizations. We have a two-way plot, a bootstrap diagram, etc. But I think the thing that I want to highlight here is you have the ability to highlight drugs, microRNAs, and SNPs directly on the pathway. Now, this particular pathway doesn't have any drugs that are known to act on it, but you'll get a sense of how this works through the microRNA modeling here. So if you remember our microRNA from our predictive microRNA analysis, we had that MIR542. We can choose that, and then it'll highlight its target on the pathway. And in this case, excuse me, in this case, it happens to target the tubulin complex, which is the source of the perturbation. And I think that adds validation to this type of analysis. Not only were we able to quickly identify a plausible microRNA that may be active, but we can also identify the most significant pathway and the source of the perturbation for a putative mechanism in that particular pathway. So very quickly we've distilled this down to something that you can take and possibly put into a paper and identify these things, or go on to your next experiment. Similarly, we can also model microRNAs that may be active in the system. You can see I choose the first one here, and it's highlighting a different target. If I wanted to narrow my search, I could choose this particular gene in here, and it has automatically narrowed my list of variants that are known to affect that particular gene, and you can see now it's highlighted with a red circle around that tubulin gene as well. So that's, I think you get a sense of how that would work. If we had drugs in this, you would be able to do the same thing with drugs and then identify some things that would be of interest to your research. So let's move on to the diseases analysis. In the diseases analysis, here we have an enrichment of plausible diseases. Now remember, this is not a disease data set, so some of the results may not necessarily make sense in this particular context, but you'll get a sense of how this might apply to your research. So we use the International Classification of Diseases to look at, or to categorize these diseases. And it goes from the most general to the most specific. The color scale on the outer band is the same as the gene ontology, where the cyan or light blue is least significant and magenta is most significant. If I wanted to look at a particular area, so here we have congenital malformations, we have neoplasms, we have endocrine disorders. Maybe I wanted to just focus in on endocrine disorders. I would click on that first ring and it narrows my focus to just that area. I can zoom back out by clicking the middle, and then maybe I wanted to look at malformations, etc. When you click on that specific disease, it gives you the DE genes that were measured and a box and whisker plot associated with that, along with literature references. You have the table, very, very similar type of layout to all the other analyses. Before I forget, you can also share these analyses with anybody. You just type in their email address, And when you add them, they will have the ability to look at what you're looking at. You can prevent them from sharing it further. So maybe it's a collaborator that maybe you're working on some top secret new biology, and you didn't want them to forward it onto a competing lab or something like that. You could prevent that from going forward. But by doing that, then they will get an email. Whether they have an account or not, they'll have access to this. The only thing that they need to do is register for a free account, and they'll have all the access that you have in this application, except for the ability to edit the descriptions. That only the report owner can do that. So that's the basic of a single contrast analysis, and I've just highlighted the very tips of each of these different components. We will have future webinars where we'll go into more in-depth areas of each one of these types of things, or give specific use cases on how you would leverage this for a particular type of use case. So I did want to highlight a meta-analysis. So I'm going to go back to my dashboard here. And I'm going to pull up a meta-analysis where we have compared 4 different breast cancer types. This is also based on a public data set. So I'm going to highlight this. And you can interrogate each of the separate analyses individually. So here we have triple negative versus healthy, we have HER2 versus healthy, luminal A and luminal B, but we have this meta-analysis. And the meta-analysis, again, this is a premium feature, so this is only available either to those people that have subscriptions or to those people that have purchased analyses and would like to cross-compare them. But you can see that you're presented with a Venn diagram, and each of the areas is color-coded. So the pink area here represents the triple negative versus healthy, and I think that's pretty straightforward. And then down below you have each of the different contrasts, and here we're presented with the genes view by default, and you can click on any one of these genes and see over on the right-hand side a description about that gene and the relative change in expression. You can narrow your table down to maybe just those genes that are in common across all of your contrasts, or maybe you're just interested in those of a particular phenotype, and so you want to just zoom in on those, and the table automatically updates with just those genes in that table. So I think the power of this comes in looking at, say, pathways or ontology terms. I'll choose pathways here. So we can see uncorrected, we have 38 pathways that are in common, but let's apply an FDR correction factor. And so now we have 16 pathways that are in common, that are significantly in common across all 4 of these phenotypes. And in here we see we have triple negative, and again we have the p-value and the related pathway. We can sort this by p-value for each of the different contrasts. So here I've chosen pathways in cancer. And then on the right-hand side you get to see all of the genes that have at least one DE gene in one of the contrasts associated to it. So you can see that some of these genes, there's only one contrast has it as being significant. But here's an interesting one here. So if we look at this AR, I think that's the androgen receptor, we can click on that particular one where we see a change in expression. And we can see that it was downregulated in the triple negative versus healthy, but it's upregulated in the other types of breast cancer, but it was only significant in the luminal A. So this may serve as a potential biomarker for future analysis. And we're going to have a use case on how you would use this meta-analysis to quickly identify potential biomarkers or mechanisms that might be of interest to you. But you can see how quickly you can drill down to a specific gene that might be of interest to you. And lastly, I just wanted to show one other element in the meta-analysis, and that's on, and this is available in all of them. I'll choose gene ontology analysis in this particular case because there tends to be a lot more terms associated with it. Even when I apply, say, elim pruning, in this case, we have 66 ontology terms that are common across all 4 of these contrasts. I can look at this in a rank diagram by clicking this View Rank Diagram. I can come down here and I can see how these are changing across the different contrasts. So in this case here, if I choose this particular term, so the extracellular matrix disassembly is highly significant in HER2, luminal A, and luminal B, but is far less significant in the triple negative versus healthy. And so that might be an area worth investigation further on in your interrogation of the biology that you're studying. So that is the application in a nutshell. If you have any questions along the way, there is a little feedback button up here. If you have an issue or a feature request or just a general question, feel free to use this, and Sidra and I will get those, or any other members of our team, and we'll try and be as responsive as we possibly can to your questions and or requests. So with that, I'm going to switch back here to my slides. And I wanted to just talk about the pricing of iPathwayGuide. So we have 3 models that I think are really a great way to go forward. And this gets to our flexible licensing and pricing model that I think helps a lot of researchers save money. So we have a pay-as-you-go model, and that's where you get to submit a data set or as many data sets as you need. And each of those data sets, you're going to get a 72-hour free preview. You can click around as much as you want. The only thing that's restricted is the downloading and that printable summary. And those are great if you just have maybe a one-off type of project. And those are $495 for each report. Then we just launched this month. We have a 30-day subscription, and this provides you with an unlimited access for 30 days. Everything is unlocked, including the meta-analysis and the printable summary. And all of those data sets that you run in that 30-day period will be available to you for life, and you can continue to come back to them and use them in future analyses for, say, a meta-analysis. Maybe you've got some data now, and maybe in 6 months from now you expect to have some additional data. You can get another 30-day subscription then, and then compare those two during that period. And so that's a great way to save money. So if you have like a small project or maybe a lower volume lab. And then finally, we have annual subscriptions. These annual subscriptions are limited for a year. You can add additional users for a fairly nominal fee. We offer free upgrades during that period. So when we update our databases, you have full access to all of those updates. And these are, I think the annual subscription is great for larger labs as well as core facilities. And we do offer academic discounts and even small business discounts. So if you find yourself in one of those categories, feel free to give us a call, and we'd be happy to work with you to provide you with a quote. Not on here is we also offer enterprise subscriptions. So if you need something for a site-wide license, we'd be happy to talk with you. Or even if you have a multi-year need, maybe you've got a grant for a 3-year period or something like that, need access to this, we'd be happy to do that for you. So with that, we'll be happy to take some questions. So I'll give you a few minutes. Use the window in the GoToWebinar to submit some questions to us, and Sidra will assemble those while you're getting your questions in. I just wanted to let you know that we will be having our next webinar in this series will be on March 8th, and we're going to really dive into how do you upload data into iPathwayGuide. We'll go over some of the different format types, including CuffDiff and SciEx swath proteomics, and then how to select the DE genes to maximize your results. We'll even highlight how you can upload raw Affymetric cell files, and some of the limitations and best practices that go along with that, and even show you how you can leverage public data through the Geo2R portal. It's held in NCBI. So with that, let's do some Q&A.
Thank you, Andrew. Before I dive into the questions, I just wanted to let everyone know that a recording for this webinar will be made available, and you will receive an email with the recording for the webinar. We have quite a few questions here, and one of the first questions that we had is there was a user who was uploading data where he had pre-filtered the data. So if you can elaborate on uploading the entire list of genes and how you can specify the thresholds within the application.
Yeah, oh, that's a good question. I think the best way to demonstrate that is in the application itself. So I will pretend that we're submitting a new analysis. What I found is that a lot of core facilities and programs will provide you with just the quote-unquote DE genes, and you can sometimes specify those thresholds on that. But the difficulty with doing that is that we would like to assess the entire background. So maybe you're doing a targeted expression type of analysis where you're only measuring 1,000 genes or proteins. That's a very key thing for us to look at. So in this example here, we have 11,000 genes that were measured, and then using our thresholds, we get 214 DE genes. Now, if you have a targeted list already, I would go back to their source and try and get the entire list. Most of those pipelines have a function of getting the entire list, and then submit that entire list, and then use these thresholds to select for your DE genes. I generally recommend that you play with the fold change threshold before you play with the p-value threshold. 0.5 tends to be the standard for p-value threshold. Fold change can be a little bit more arbitrary. We've settled on 0.6, but I've seen people use 2. I've seen people use 0.3. So you can use a variety of fold change thresholds.
Going along with that, do you recommend a certain number of genes to be differentially expressed in the entire list of genes?
Not a number per se. There's no magic number, but what we have found, and this will be highlighted in our next webinar, is that generally between 5 to 10% DE genes seems to work best. So if you have a list of, say, 10,000 genes and you only have 2 DE genes, you're not going to get anything significant out of that. Similarly, if you have 10,000 genes that you submit and you try to select all of them as differentially expressed, you're not going to get anything that's going to be useful to you either. So you want to find that sweet spot, and what we have found is between 5 to 10% seems to be the sweet spot. And in our next webinar, I think we even have a citation for where that number comes from, and I'll make sure that we highlight that in the next webinar.
Another question that I have is, what kind of formats do you need from Discovery Proteomics to export the data and perform the analysis using this tool besides SciEx?
That's a good question. We've had a lot of requests. Our relationship with SciEx is an exclusive relationship. So we probably are not going to offer additional proteomics importers per se, but there are ways that you can do that through this custom tab delimited file. The only thing on there is that you will need to translate your protein IDs to gene symbols, to official gene symbols. So if you have, say, UniProt IDs, then there are a number of translators out there. If you want to send us a question, I can certainly send you a link to the translator that I have found to be most useful. But if you have official gene symbol, fold change, and p-value, you should be ready to rock and roll, whether it's from transcriptomics, RT-PCR, or targeted expression, or proteomics.
Thanks, Andrew. Another question is, what is your plan to include more species?
The squeaky wheel gets the oil. If there's a lot of demand for a specific organism, we would be happy to look at it. I know we've had requests to add zebrafish and some other model organisms. I think one thing that has one of our customers, that what they do is they use homologs and do the translation to homologs. I think that's the best way, and I know that there are other applications out there that suggest to do the same thing. We've talked about incorporating a homolog translator in the future, but there's a lot of other requests that seem to have a little bit of higher priority right now. But hey, the more you ask for it, the more likely you are to get it.
Great. I'm actually going to combine two of the questions because they are similar. One of the questions was, can this tool be used for RNA-seq analysis? And another question was about someone who wanted to identify biomarkers based on variant data. and she wanted to know if she could upload variant data.
Gotcha. So iPathwayGuide is currently designed for expression, protein or gene expression, or transcriptomic expression. So that would be your RNA-seq, your microarrays, as well as your mass spec discovery proteomics type of data. We do not currently support DNA sequencing, but we are in the final stages of putting a beta together for a new application called iVariantGuide that will accept VCF files and will be able to provide you with an interpretation of pathways that are likely impacted based on that variant data. And I think you can probably put the breadcrumbs together, and you can see in our pathway analysis we have that section where you can model the SNPs or the variants directly on the pathways. We will eventually build that bridge across both the gene expression as well as the DNA analysis pipelines.
Thanks, Andrew. Another question is the KEGG database the only pathway database used by iPathway? What about WikiPathway, Reactome, etc.?
That's another really good question. Yes, currently the only pathway database that we're currently using is the KEGG database. We find that it still delivers really good results because it's conservatively curated, but we do have it in our books. As soon as our beta for our variant application comes out, we will be looking at adding Reactome pathways, and also have the ability to possibly do network analysis based on those different databases.
Thanks. Another question is the corrected p-value when less than 1, is it still significant or not? And moreover, when the digits are shown in black bold compared to green ones, what does that mean?
Oh, so that's a good one. Let me go back here to the application, and I think that's probably the best one to show is in this meta-analysis. So the p-value significance, I mean, it's a range from 0 to 1, obviously. Whoops. So in the actual analysis, when you look at it, so in this genes table, the green is highlighted because it is significant. It's in bold. And then we have the fold change that's also significant. And by default, we're only showing the significant genes. If I chose to show all genes, and I'm not going to do it here because it's going to load up 20,000 genes here in this case, and it'll take forever for us to do it on this webinar, you would get a list of all those genes, including the ones that were not significant, and then you would see values down here that are not bolded and not green. So I think that answers that question.
Yes, I think so. Another question is about the methodology. Does the pathway analysis take into account direction of the gene, whether it's up or down regulated, for consistency on the enrichment of the pathways?
Yes, that's a big, bold yes. So not only do we look at the, let me see if I can find that slide here very briefly. Yeah, so we do look at the direction as well as the type of relationship, so whether that's activation through phosphorylation or inhibition or all that kind of stuff. So yes, we do take into account not only the direction, but also the signal change between the genes on the pathway.
Thank you, Andrew. Another question is about the Affymetrix cell files. The Affymetrix cell file platforms that we support, is it similar to just downloading the data from Geo to R and uploading that?
It is similar. We don't support all the platforms, all the Affy platforms. If you look on our FAQ, you'll see a list of the platforms that we currently support. We are looking to expand that to additional platforms. But yes, you can, if the platform is supported, you can actually download the raw cell files from Geo to R and then upload them. The only caveat is that the comparison in your control group must be of the same platform. You can't have different platforms between those two groups. That's the main thing. And then we'll do the QA I'm sorry, the QC as well as the normalization. And we're going to have that in our next webinar. We'll go into greater detail on how that works. But there is a video out there. In fact, I think it went out today. You might have even gotten an email from us on how to upload data. I believe there is a link in that email that tells you how to upload AFI cell files.
One of the last questions, can a text or Excel file be uploaded?
So a text file, yes, but not an Excel file. Where did I have that? I'm going the wrong way here. So here it is. So what you would want to do if you have a text file, just make sure if you're using Excel, make sure you save it as a custom or as a tab delimited file with the extension TXT on it, and then use the custom tab delimited uploader in iPathwayGuide, and then you'll just have to choose which column is your column for your gene symbol, and that should be the official gene symbol. I'll just reiterate that again. The log fold change, not just the raw fold change. We want the log fold change, and it doesn't matter whether it's log 2 or log 10. That's kind of up to you. And then the p-value and or corrected p-value if you have that. Most of the pipelines that get you to differential expression will have all of these three components in it. So it's just a matter of saving it in that format and then uploading it into iPathwayGuide.
We are actually running out of time, but one of the attendees said that this is a great tool. How long have you guys been working on this?
So that's a really good question. So we got this grant in, I think it was 2011, and we spent the first couple years perfecting the analyses. We had a desktop application. We made a lot of mistakes on how to deliver software, and then we figured out the right way to do it. And then we went commercial with this application just over a year ago. And we have nearly 6,000 registered users now, which is, I think, a great testament to the usefulness of the application. And so, yeah, we've been around for quite a while, and we have some nice partnerships that are out there. And I think you'll be seeing more and more of us as time goes on.
All right. Well, this is all for questions. Thank you so much for joining us. If there are a few questions that we didn't get to, and me or Andrew will make sure to answer those questions. We'll probably end up sending you an email. As I mentioned earlier, recording of the webinar will be made available, and you will get an email with a link for the recording. And also, Andrew has already announced the second webinar, which might be of interest to quite a few of you guys because some of the questions were about file formats and how to upload those file formats in iPathwayGuide.
Okay.
And that's it for now. Thank you for joining us. We hope to see you on March 8th. And also, if you have any suggestions on the topics for webinars, please feel free to let us know.
Yeah, here's our contact information. You can either call us or send an email to info. Both Sidra and I will get that, and we'll be sure to incorporate that into future webinars. We'd love to hear from you if you have a specific use case, or if you have some data and would like us to maybe use that as a use case. We're always open to those kinds of things. Obviously, we'll have to pick and choose those that work best for everybody, are most representative to the widest number of people. But other than that, thank you, everybody, and we look forward to seeing you online and in future webinars.